Well, let's get started for today. So this is going to be our second lecture, and we're underway. And today, what I'm going to talk about the topic of today is data. The reason I hesitated there is I'm trying to, my mind is dealing with the fact that data is the plural form of datum. And so in this class, I'd like us to aspire to talk about the data are complex, or the data are visualized using this plotting. And but in fact, there's a disagreement about whether data can be used as a singular. But I'll be using it as the plural throughout this this course. So today I'm going to talk about, we're just going to talk about data. And that's really our introduction to statistics and genetics. And we're going to talk about some real data that I have collected from you over the past couple of days. And today I want to talk about real fundamentals, and we're going to go through pretty systematically talking about how we visualize data, how we describe data. We'll talk about concepts that you may be familiar with, measuring the central tendency of the data, using methods that you may have learned as early as middle school or high school, things like the mean and the median and the mode. But then we're going to talk about dispersion, how we talk about the spread in the data, and we'll talk about the concept of location. And today, a topic that may be new to you, but one that's really important to understand is the degrees of freedom that our data have. And so I'll try and convey that concept. And then today, for probably the only time in this course, I will show you a demo of me using R. And so this is just because many of you are new to this, and I want to show you how it's useful to see different people interacting with the R programming environment. And I want to show you how I find my way around and use R and RStudio. And just because it's good to see different people using it, and I'll also be showing you how to make plots in R, which is something we're going to do. We're going to do in the assignment. Okay, so that is going to be very exciting because it's going to be live in real time. There's no guarantee that things won't go terribly wrong, but that is what we're going to do today. So let's get started here and just remind ourselves about statistics. What is statistics? Statistics is simply the science of collecting, organizing, and interpreting data. And so that's what we're going to aspire to do today. And our goal today is going to, not necessarily in this order, but we're going to talk about how we get data, how we visualize those data, and how we describe the data. And I'm going to do this in real time using typically, I would be writing on the board, and in this case, I am going to write on my iPad, and we'll see how that works. So I'll stop sharing that, and I will share my iPad via AirPlay, and let's see how that goes. A plugin is required, always. Right, now I'm on it. I'm going to tap my screen mirroring. I'm going to go tap zoom, and whammo. Do you see a table on your screen?
Yes.
Yes. Yeah. Great. All right. So I'm going to talk first about something that's not a heritable trait, but something that's near and dear probably to many of your hearts, and that's your GPA, grade point average. Okay. And so here I've collected some data, and I've put this in the format of a frequency table. And so in this table, you can see here that I've got the GPA, so numbers like 3.3, 3.4 going up incrementally, and then the frequency in a class of the number of individuals who have that GPA. So 4 individuals have a GPA of 3.3, 5 individuals have a GPA of 3.4, and so on. Okay. So I'm going to write, you can, if you want, you can replicate this. I will also, everything I write here, I'm going to export as a PDF at the end, and you can you'll have a copy of this as well. But here's our little table. I'll make it smaller so you can see. Okay, so what's maybe the first thing we need to know? Well, one important thing that we should know here is what is our total sample size. So we're dealing with a finite number of individuals, and we've recorded their GPA. So let's count them up and let's see. We've got do some basic arithmetic just to get ourselves warmed up on a Tuesday morning. 9, 19, 20, 22, 23 individuals. So n equals 23 here is the total number of individuals. And so if these are our data, the question that I want to ask is what is the best way to convey this information visually? Who has a suggestion?
Couldn't you do a frequency plot or a histogram?
Right. So that would be, I think, a really good way of being able to present these data. So often, you know, the human mind likes to see pictures rather than tables. So we look at this table and we form maybe in our mind a picture of what these data look like. But it's much better when we're presenting information to be able to convey that in a visual way. So one way we could present this information is as a histogram, as Justin said. Another way would be some sort of scatter plot where we plot the data. So let me show you what a scatter plot might look like. So we could, for instance, we could make a y-axis scale here that has the GPA on it. We could indicate the possible values. So we'll start at 0 here. The next value in our table is a 3.3. And so we're not really interested in values below that. So often what we might want to do is break the axis, which we indicate like this to indicate that there's some missing values. And we're really starting at 3.2 here. And then we could indicate the possible values, 3.4, 3.5, 3.6, 3.7, 3.8, 3.9, 3.10. That's 4, right? Yeah, 4.0. And now we could simply plot or indicate at each of these points for our individuals the number of individuals that have that value. So for example, if we look at our table, we see that 4 individuals have a 3.3. So in that case, we could just put 1, 2, 3, 4 points here. Let's see, what do we have? 5 individuals have a 3.4. 1, 2, 3, 4, 5. We have 10 individuals who have a 3.5. So now we need to put 10 points here, 3, 4, 5, 6, 7, 8, 9, 10. No individuals have a 3.6 or a 3.7. We have 1 individual who has a 3.8, and we have 2 individuals with a 3.9 and 1 individual with a 4.0. Okay, so now we have a type of scatter plot that indicates and visually depicts the data. And it has, you know, some virtues, but this is certainly one way of being able to convey these data. Another intuitive way to be able to show these data is to be able to plot a histogram. And so what would a histogram do? A histogram, in this case, we would plot a 2-dimensional plot. We would have a y-axis and an x-axis here. And on our y-axis, in this case, we will put frequency, the number of individuals. And on our x-axis, we will put the GPA. Okay, and so let's again, if we start at 0 here, we're going to break this because we're going to ignore values below, for example, 3.2. and then we'll have 3.3, 3.4, 3.5, 3.6, 3.7. Okay, and then we'll plot our frequency. And so we want the range and our frequency to reflect the range of the data. So we're going to go up to a value of 10 in this case. So we need to put 10 breaks here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And now what we're going to do is we're going to plot for each class, each numerical value of the GPA, we're going to plot the number of individuals. So we see that the number of individuals, again, with a GPA of 3.3, and in this case is 4. And in this case, I'm going to draw it like this. So we now can look at this and see immediately that there are 4 individuals. For the 3.4 GPA, so it would look something like this. 3.5 is where we had 10 individuals. So now we're going to have a big bar that goes all the way up to 10 here. Then we have 3.6 was 0, 3.7 is 0. We have 1 individual here and 2 individuals with a 3.9 and 1 individual with a 4. And now we've just plotted a histogram. What are the relative merits of each of these plots? If you were to, you have to make a decision as a an author on a scientific paper or a medical paper or some sort of report that you're writing. You have to make a decision about how you're going to convey your data. Which of these methods of conveying the data is, what are the relative merits of them? Or maybe let's just do a thumbs up. Who likes the scatter plot? If you do a virtual thumbs up. Doesn't look like too many votes. Who likes the histogram? I see some thumbs going up. All right. And what maybe is one major advantage of the histogram over the scattergram or the scatter plot that I've shown?
I think on the histogram. Oh, go ahead.
No, go ahead, Avni.
Oh, I was going to say, I think the relative sizes are shown more clearly on the histogram than in the scatter plot.
Right, yeah, so it's easier to see the difference between the different GPA scores, perhaps, than looking at these bars rather than looking at the counting up the points. What's another advantage?
It's much easier to see the actual value for frequency on the histogram than the scatter plot because of the y-axis labels.
Right. So you have to do less work as a viewer of these data in order to interpret it, right? So in the scatter plot, we have to do the mental work of looking at 3.5 and saying, how many are there? And you have to actually count them in your mind. Whereas we can look directly at the histogram and we can count, we can easily read off the histogram, there are 10 individuals with a GPA of 3.5. So it's an easier way of being able to see these data. In doing this, we've made a decision, and I just want to point this out, is that here, what we've done is we have what's called bin the data into bins of 0.1. So we go between 3.2 and 3.3, between 3.3 and 3.4, for example. But we could change the bin size. So what if we change the bin size? Or the break size. In that case, we could make a different plot, something that looks like this, again with the GPA on the x-axis and the frequency on the y-axis. But in this case, we could make our bins so that, for example, they go between 3.2 to 3.4 to 3.6 to 3.8 to 4.0. And now if we add up those individuals that have a GPA between 3.3 and 3.2 and 3.4, for example, those are all the individuals that have a GPA of 3.3 and 3.4. So that's 9 individuals. So now in this case, our plot would look like this. And if we look now at the next class between 3.4 and 3.6, there's only 10 individuals. So now we would make a plot that looks like this. And finally, between 3.6 and 3.8, we only have 1 individual. And then we have 3 individuals in the next class. Okay, so this is just to show you that we have exactly the same data, but the plot looks totally different to this plot here. Okay. And that's because we've made a decision to reduce the resolution of our plot. And so this might have advantages in some cases. In other cases, it might have, one might argue that this actually is less informative because we don't have the same sort of resolution. But these are also questions we need to ask ourselves when we're making these sort of plots, is what is the level of resolution that's necessary to convey the information that I want to be able to convey. So there's 3 fundamental rules of visualization that I just want to state here when we're visualizing data. So one key thing always is that your axes must be labeled. This is something that you want to learn at a very early stage. Even if your y-axis is frequency or count, it always must have a label. Otherwise, you're asking the person who's reading your plot to guess what it is. And so both axes must always be labeled. And you can see here that I've labeled both the GPA and the frequency, and that is correct. Typically, what we also want for the axis label are the units, an indication of what the units are. So, for example, if we're plotting height, we want to indicate whether it's centimeters or inches or feet. So we want to indicate that as well. You need to use the visualization that best illustrates your point. And this is very much the art of data presentation and trying to think of what's the point I want to make and what's the best way of being able to convey that. And then a final point is you want to think about the concept of ink versus information. This is an idea that comes from Edward Tufte, who is a master of visualization, has written many, many books about visualizing data. And the idea here is simply that you want to maximize the ink to information ratio, okay? And so you don't want to put any unnecessary, you want to think of ink, which in this case is printer ink, as a limited resource, and you want to use as little of it as possible, or only as much as you need. So one key example of that is that it's much better to draw a histogram that looks like this than to think, well, I'm going to make my histogram a 3-dimensional histogram that looks like this, which adds no additional information, but uses up additional ink. And typically when we're visualizing data, we much prefer to be able to keep our plots simple and not use any additional features of the plotting that are unnecessary. All right, so moving on now from visualization, now I want to talk about descriptive statistics, how we can describe the data. So okay, and the first class of descriptive statistics I want to talk about are the central tendency of the data. So there are three ways that we typically do this, and they all begin with the letter M. Who can give me one?
Mean.
Mean, right.
Median.
The mean, the
Mode.
median,
Mode.
and
Mode.
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 Right.
Mode.
And when was the first time you ever learned to calculate the mean in your life? How old were you?
Third grade.
Third grade? Yeah, okay.
Fourth grade.
So it turns out it's very important. And so we'll revisit it here. Let's remind ourselves how to compute the mean, which is also the average, is the synonym for it. So in this class, I'm going to try and minimize the amount of mathematical notation that we'll use. I find it a bit of an impediment to understanding when you are in a class and people are talking about the ith row and the jth value. But sometimes some mathematical symbols are important for being able to summarize information. So here's an example where we'll define the mean. We'll use x bar, so with a bar over the top of it. And all we're going to do to compute the mean is we're going to take every single value of x that it can take. So in this case, that's every single GPA value. We're going to add them all up. And so this is the symbol we'll use to add. And then we're going to divide that by the total number n. Okay, so this is just, here we'll define this as n is the number of samples we have, and xi is each individual value. All right, so if we go now to our table up here, why don't we maybe for the first time since third grade calculate the average GPA by hand using pencil and paper or tablet and stylus if you have it. And then once you have what you think is the right value, let's pop it in the chat and see how they compare. All right, looks like we can all still do arithmetic. So 3.5, I think, is the correct estimate of the mean. And you can see here that, we had to look at this table and we had 4 values of 3.3. So I've just multiplied 4 times 3.3. And so this is, and so on, 5 times the 3.4 value added up, or 23 values and divided by 23. Okay, so we have the mean. We have the mode. Who remembers what the mode is?
The number that occurs the most often.
The value that occurs the most frequently. Great. And so there is no mathematical formula for computing the mode, or we don't really need one. We can just look and what is the modal value for our GPA?
3.5.
3.5 is the most common number. And actually, that's one of the advantages of these ways of plotting. We can simply look at this and we can see, oh, indeed, here's our mode, here's our most common number. And again, we can see it here. And you can see in this case, we've lost that resolution. We don't know what the mode of the data are by plotting it with this number of breaks. All right, and finally, the median is our third measure. And who remembers what the median is?
It's the number that appears in the middle of the data set.
When we do what to the data set?
When you order it from least to greatest.
Right. So the median is the middle value when the data are arranged in increasing order. Okay, so we have 23 values here. What's our middle value?
3.5.
Not the middle value. Which, if we ranked them, what is the rank of the middle value? Sorry. So if we have 23, so you can have rank 1, rank 2, rank 3, rank 4. So 1 through 23, we put them in either, we can put them in increasing or decreasing order, but in some rank order. And how do we, if we count from 1 to 23, what's the middle value of along that number line? The 12th.
12th.
The 12th value, okay. What if we lost one of our school members? They decided school wasn't for them. Too easy, they're at 4.0. Now we have 22 students. What's the middle value now?
We'd average 11 and 12.
We would average the 11th and 12th, right? Okay, so there's a little rule then that we can use, which is the following. So if n is odd, so the number is odd, then the median is the n plus 1 divided by 2 position. So in this case, that's 23 plus 1 divided by 2 is 12. If n is even, it's the average of the n divided by 2 and n divided by 2 plus 1 position. So if we had 22, it would be the average between the 11th and the 12th position. Yeah. This is a little preview of your future where we're making if statements. So we have if n is odd, we do one thing. If n is even, we do a different thing. And so this is something we'll do in computing. Often we'll write little computer codes where we'll specify different computations depending on the value. And this is what we're doing when we're computing the median. So for n equals 23, it's the 23 plus 1 divided by 2 value, which equals the 12th, and therefore it's 3.5. Okay, so for our data, we have the mean of 3.51, we have the mode of 3.5, and we have the median of 3.5. So they're almost the same, but not quite the same. Okay, so these are 3 distinct measures, and that depends on your data. And so the key thing to remember is that the mean is not equal to the median, and it's not equal to the mode necessarily. And so we need to compute each of those independently. And what these values describe is the central tendency of the data. where do the data tend to be the highest density? And so they provide three. That's a useful way of being able to summarize the data, is talking about the central tendency. What about the spread in the data? And so when we talk about spread, one of the measures, so if we're talking about dispersion, how dispersed are the data? And so one of the useful metrics we'd like to use is the sample variance. And what does the sample variance do? The sample variance measures the distance, or it measures the distance of each value from the mean. And then we determine the average distance across all the values. And some of those values are going to be negative and some of those values are going to be positive. And so the way that we do this to compute this value is we're going to take each individual measurement, xi, so that's each GPA score. We're going to subtract the mean, which is our x bar, which we computed, which is 3.51 in this case. And we're going to add up all those measurements. We're going to do that, and so I'll use the nomenclature, which I may not have put on my mean. So from the first value to the last value, which is the nth value for 23, that's going to be all 23 values. Now, some values are below the mean, some values are above the mean. So if we did this, if we just added up all these differences, we'd get some of these values would be negative and some would be positive. And so we'd get a value that is somewhere probably around 0. So what can we do to mitigate that problem? Well, we can turn them all into positive values by squaring this difference. And so what we're going to compute here is called this metric on the top. If we add them all up, this is called the sum of squares. And it's the sum of the squared difference between each value and the mean. And we're adding up all of them. And now we want to know the average of the sum of squares. So how do we compute the average from our data? We just divide it by the number of data points, n, was going to give us the average. But, and here's where this concept is going to become important, we need to do one thing. We need to account for the fact that we have lost a degree of freedom in our data. So we're actually going to subtract 1. And this is our degrees of freedom. By the end of this class, you're going to understand what degrees of freedom is. This is a very important concept in statistics that lots of people don't understand, lots of people talk about, but don't actually understand. But I think I have a way of being able to convey to you what it means, and you understand by the end of this. Okay, so we're computing now our sample variance, and the symbol we use for this is S, and we squared. And so this is our equation now for the sample variance.
Is it the same as standard deviation?
It's not the same as the standard deviation. Is the answer. We'll come to the standard deviation. But here, okay, let's take 5 minutes now. And maybe for the first time in your life, compute the variance of this data set by hand. So we're going to use this formula. And what do we need to do? Well, the first thing we need to do is compute the sum of squares. So that's we need to take each value and subtract the mean. So I'll get you started in this case. S squared. Our first value is 3.3. We're going to subtract the mean, 3.51, and we're going to square that. And we're going to multiply that by 4 because there are 4 individuals that have a mean of 3.3. The next thing we're going to do is go to our next value, 3.4 minus 3.51. We're going to square it. And how many of those do we have? 5, and so on. So I'll give you until 11:45 Eastern Standard Time to see if you can compute the sum of squares and then the variance. I'll disconnect mine and do it in real time as well. We'll see if we get to the right answer. All right, I see some answers coming through. It looks like a consensus. Has anyone ever calculated the variance by hand before? I see some nods. Hopefully it's the first time for many of you, because it's today, it will be a very memorable day for the first time that you ever had to calculate the variance by hand. I'll provide you with a couple more opportunities in this class to be able to do this, but this is part of learning or appreciating the value of using R in statistical programming, because the computer's going to be able to do this for you. But let me connect my iPad here again. Right. All right. So you can see now in this case, I have to do this whole calculation, add all these numbers together, divide by the denominator, and I got 0.0394, which I think a number of you got. And so I added everything up on the top. What's the numerator called in this case?
The sum of squares.
Sum of squares.
That is the sum of squares, okay? So that's an important metric or quantity to be able to describe. So that's our sum of squares. And our denominator, what is the denominator? The degrees of freedom. Okay, and so that's how we calculate the variance, is taking the sum of squares, so the sum of squared differences of each of the values, subtracted from the mean, squared them so that negative values become positive, add them all up, divide by the degrees of freedom, we get 0.0394. So 0.0394 is the variance of our GPA data. And what are the units of this value? So we're talking about GPA. So when we calculate the mean, we can say there's a mean of 3.51 GPA. And what are the units of the variance?
GPA squared.
GPA squared, right. So it's 0.0394 GPA squared. Do you have a good intuitive sense of what GPA squared means? Not really.
Not really.
Yeah, no, it doesn't really make sense, does it? So we'd like to maybe put it back into units that are useful to us. And so what we could do then in that case is we could say, well, let's take 0.0394. And what if we take the square root of that? Then we're going to get back to units of GPA, right? And if we do that, now we are going to have a value of 0.198. And that is the value that we call the standard deviation, which is the square root of the variance. Okay, so now we can say, and sometimes we just use the value S because we have used S squared for the variance. So the standard deviation we can just summarize as S, and that has units of GPA. And so that might be useful then. So one way to be able to summarize both the central tendency and the dispersion would be to say that, for example, we could say the mean of the data plus or minus 1 standard deviation is equal to 3.51 plus or minus 0.198. Would be a way to be able to summarize and report both the central tendency of the data and the amount of spread in the data. Just while we're here, we can talk about another interesting metric just for your completion, is that we can talk about, we can get a measure of the relative variability within a data set. And we can compute a value which is called the coefficient of variation. And that is simply CV is equal to our standard deviation divided by the mean. And then often we can multiply this by 100%. So in this case, our standard deviation is 0.198. The mean is 3.51 multiplied by 100%. And now our coefficient of variation is 5.64%. And this value is useful for comparing variation between data with different means. So for example, maybe you wanted to compare the amount of variation in this class to a class in the UK where they only have GPAs that go up to 3. And so they're going to have a different mean, but you'd like to be able to talk about the variation among the class. This would be a way of being able to compare that. So that's a useful metric to have. All right, I want to talk about location. So when we're talking about any specific values, this is another way of sort of talking about the variability of the data. And this is the concept of percentiles. And this is really just a terminology. You're probably all familiar with it. You all, I think when you do your SATs, don't you talk about what percentile you are? Did anyone, what percentile did you get for your SATs? You can lie. No one's going to check.
99.
99, yeah, of course. We all got 99th percentile, right? What does that mean? That means that, does that mean that 99% of people did better than you or worse than you?
Worse.
Worse, right. So the 99th percentile means 99% of the data are less than or equal to that value. And 1% are better, okay? And so the reason I want to introduce this is because this is useful in a type of plotting that we'll look at. And some useful values often are the quartiles. So we can talk about the 25th percentile, the 50th percentile, and the 75th percentile. And this is called the first quartile. This is the third quartile. And one terminology that's used is the interquartile distance, is the distance between these two values. Does anyone know what the 50th percentile is equal to? So the value at which 50% of the data are less than and 50% of the data are greater than? Median, question mark. Median is correct. The 50th percentile is the median value. Now, the reason that this is useful is because another type of plot that we could use is called a box plot. And this is a plot that we'll use in R. And here, in this case, we would have GPA, but we can aggregate the data to summarize it, and we can make a plot that looks like this. And it's going to have this line and bars that look like this. And this is our median. And this is the 25th percentile. And this is the 75th percentile in a box plot. All right, now before I want to have time to be able to give you a demo of me using R and being able to make plots like this in R and work my way around it. But before I do that, I want to cover the concept of degrees of freedom. Okay, and the degrees of freedom are literally the number of independent pieces of information that go into the estimate of a parameter. Okay, so when we estimate the mean, that is a parameter. When we estimate the variance, that is a parameter. And so the degrees of freedom are the number of independent pieces of info that go into the estimate of a parameter. So let's consider the following scenario. Let's say we have 5 numbers. I'm going to give you these 5 numbers here. 1, 2, 3, 4. 1, 2, 3, 4, 5. Here are 5 numbers. Okay, these are we have 5 numbers. So n is 5, and we have with a mean, we have estimated the mean, and the mean is of 4. Okay, so the mean of these 5 numbers is 4. If we know that, then we must know what the sum of the data is. What is the sum? We have 5 numbers. They have an average or a mean of value of 4. Therefore, the sum of those 5 numbers must be, sorry?
20.
20, right? Sum must be 20. All right, now in the chat, first value, give me one possible value that could have gone right here. 1, we'll put a 1 right there, okay. Very good. Now in the chat, give me one additional, what number could be our second value? Let's think creatively, people. 1 is the loneliest number. Let's get a very exciting number, 6. That's getting a bit more exciting, not super exciting. 6 was first, so okay, now okay. Can we get a bit more creative? 10, okay, we'll put that here. Think big. -8, aha. Okay, now what value can go here? In the chat, give me a possible value here where we know this fact. But I want you to be creative, right? It has to conform to these rules, but we're being as creative as possible. But your creativity is being restricted by reality, and it must be, and I'm going to put that in red, it's 11. It cannot be anything else. And why is that? Because it must add up to 20, because we have computed the mean of 4. So by computing the mean, we've constrained the data such that we lost 1 degree of freedom, okay? And so the degrees of freedom, and this is something I want you to remember for the rest of your life, the degrees of freedom is the sample size n minus the number of parameters p estimated from the data. Okay, n is our sample size. In this case, it was 5. P is the number of parameters we've estimated from the data. In this case, it was 1 because we've estimated the mean. Therefore, we lost 1 degree of freedom. Okay, and so this is important to remember because when we measure the variance, which we said is s squared, that's equal to the sum of squares divided by the degrees of freedom. I'm going to use that shorthand, which is equal to the sum of all the squared values. I'm going to keep you on your toes by using y now instead of x, just as my variable. We're going to add them all up. We're going to divide by n minus 1. When we do this, the first thing we do is we add them all up, add up the difference from the mean. So in that case, we had to estimate this mean, and this is calculated from the data. So we lose 1 degree of freedom, okay? And so this is the concept. We'll come back, we'll keep learning about degrees of freedom, but it's really important in statistics as a concept. It's often something that people don't really understand, but it's literally the freedom of your data, and you're constricting that freedom when you estimate parameters from the data, okay? All right. So we've been through ways of visualizing data, ways of summarizing both the central tendency and the dispersion of the data, degrees of freedom. These are concepts we're going to keep covering in this class. But now what I would like to do is show you how we can do this not by hand, but do this in the computer using R. Okay, so I'll stop sharing here. And I can, are there any questions? I see all the 11s, that's good, in the chat. Has anyone ever learned about degrees of freedom before? That wasn't covered in 3rd grade, right? All right. I'm going to try to use R in public, okay? So I don't do this very often, but we'll see how it goes. And what I want to do is I'm going to show you, this is like, you know, looking under the hood at the reality of what computer programming is and statistical programming. And the reason I want to show you is because, you know, it's hard, you make lots of mistakes. So I'm going to show you all the mistakes that you make along the way and all the little decisions you need to make. And so to do that, I asked you to put your height in a what turns out to be a non-anonymous way of being able to insert your height, your own parameter, your own values into a spreadsheet. And that's human height is a quantitative phenotype, and that is that it varies continuously in the human population. And so we can ask in our sample we can ask these questions about what is the mean, average height in our human genetics and biostatistics class. We can plot the data. We can ask about the variability. And so let's do that. Okay, so let's see. First I want to go to my Google Drive document, and you can follow this in real time and do it along with me if you're able to. Okay, so let's go first to wait, let me go here. Let me stop that. Okay, I'm going to go to where is that? here. Okay, are we looking at this spreadsheet where you all filled it out for me? Okay,
Yes.
and that's good. Okay, now I'm grateful that you made it complicated for me. So that's, you know, getting data, it's non-trivial. Okay, that's a lot of what we do as scientists, as doctors, as, you know, getting the data and getting it in the right format. So here I can see this person did not put a number here. So for today's purpose, let's just make up a number. I think 173 sounds good here. So incomplete data is a big problem. And so I'm just going to make this easy for us by guessing this person's a male, this person's a female, this person's a male. Let's make some up. I love this person, how precise they are. That's great. Thank you. And this one too. And then down here, we don't have any values. So I'm just going to delete them. Okay. So here we go. I think everything looks good. Now what I want to do is I'm going to download this as a file, a text file that can be read into R. So to do that, I'll go to download, and I'll download it as either a comma separated values or as tab separated values. Why don't I do it as tabs? Okay, now you can see down here that this has been downloaded. And here's a key thing that maybe many of you haven't had to worry about before, which is where are the files on my computer? That turns out to be really important, is knowing where they are. So in this case, on a Mac, I can say show in Finder, okay? Now I can find that. And so whatever you've done on your computer, you need to know, where did I download it, okay? And because now I'm gonna take that file and I'm gonna put it into the relevant place. So I'm doing that right now. You're not seeing that. But I've put it somewhere that I know it. Okay, so I just want you all to get in the practice. Maybe you're used to downloading things and they just get into your download folder or your desktop or something, but you don't want to be, everyone here has got grandparents who say, you know, I downloaded this thing and I can't find it on my computer, right? Don't be your grandparents, be a computer scientist who knows where things are on your computer. And why is that important? Now that's important because I'm going to go to my studio, which is right here. And let's see. Now, when I'm in our studio, so we're looking, I was trying to make sure some things were working before, but I will we'll ignore that right now. Okay, so on our studio here, now I'm in my R studio, and what I want to do is I want to read those data into my current, where I currently am. So the first thing I need to know is where I am. So this little window over here allows me to click around and find where I am. And in this case, I know I move that file into here, into Google Drive, into my drive. I have a folder that's called NYU Teaching. Here it is. I have a class that's called Human Genetics. Here it is. It's 2021. It's lecture 2. And I moved it right here. This is, does everyone see the file that I put right here? Heights-sheet1.tsv, okay? That's the data, and it's in the place that I want to be. Now I just need to get R into that place, and that is often a challenge as well. So I'm gonna clean this screen up so you're not looking at anything that was there before. And now here I'm in my terminal. This is called the terminal window over here. And one thing I'd like to know is, where am I? Or I want to put myself in this directory. And one easy way to do this in our studio is to go here, click on more, and say set as working directory. And it's literally going to type the command here that's going to put me in this directory. And I can see what's in this directory by doing something like ls, which means list. And it's going to tell me, no, sorry, that's not correct. Just ignore that. I can see what's in this directory by looking over here. This is telling me what data and what different files are in this directory that I'm in. If I want to ask what directory I'm in, I do get wd, get my working directory. And every time in R, you put these brackets, and that indicates a function where we can put an argument or we can put nothing in this case. And it tells me this is where I am. So great, I'm in the right spot. And now what I want to do is I want to read those data into my current R existence. And one easy way to do that is to go up here to this window and import data, import from a text file, because I made it a text file, and choose that sheet. There's the .tsv file, and I'm going to say open, and it's going to pop up a little window, and it's going to say, do you want to open these data? Is this what you want to open? And here I can rename it. So I'm going to call it, in this case, student heights, and I'm going to use what's called camel case. I guess maybe we can start with a lower and write it like this, no spaces. So this is actually the name of my variable, which is this data sheet that I'm going to read in. And then let's look at this. Do I have a heading? Yes, I do have a heading. I have heading student height and sex. So yes, they're separated by tabs. It could have been separated by something else, but in this case, I'm going to choose tabs. And then everything else I can not worry about at this point. But this gives you an idea of what the data are going to look like when I read them in. And so now I am going to press import, and it's going to import it. And in this case, it automatically pops up a little viewing window, which allows you to see the data frame up here. Okay. And so what this did is it made a variable called student heights. That's the name of the variable here. And that is what I want to, if I type that, it's going to print it out here in this console. So there's two different ways of interacting with the R programming environment. One is in what's called the console here. But the problem is once I close this all down, this is all going to go away and not be saved. And that is why what we want to do in this case is we want to go to and open a new R notebook. And that's what we're doing up the top here. And in this case, we can, this is called our YAML. This is going to specify what it's doing. And we can edit all of this. The format here is that everything in gray, this is computer code, and everything that's outside of the gray is just plain text. And so we can edit this, and we have this YAML at the top. So I can change this and say author is David Gresham, for example. And now I can edit all of this, and I can say this is David Gresham's Markdown, and this is all editable, right? Now what I want to do is I want to type my commands in here in what's called this chunk.
Professor?
Yeah.
Just a quick question. Back to the import file, how did you get there?
So let me show you again. So we'll go to console, and I imported the data using this command. So this is the command here. It's going to make student heights be the variable name, and it's going to read it in using this function called read delim. But to do that, because you may not remember how to do that exactly, there's a little interface that allows you to do that. So we can go up here to import data set. I need to get this out of the way. From text base, and I choose that, and it specify my name here that I want to call it. Okay.
Okay, thank you.
Let me call it something different in this case, students. I'm going to import, and now you can see all that it did is it printed out the command that I could have written myself, but I'm lazy, so I just did it that way. But this command is actually useful, and I can just copy it now from here and go back to my R Markdown and actually put it in, copy it here. And now what, if I run this chunk, it's going to actually run it here. I had nothing, there's no output, but that is going to have read it in. And now I have a little, now I can tell myself, what am I doing here? First, I am reading in the data. And that's the first thing I'm doing. Okay. So the point is, when I use R, I mess around down here. This is how I sort of explore things. And then I copy and paste things that work up into this R Markdown document. That might be one way to do it. Okay. And then here, what I want to do now is I want to say, okay, what can I do now? Well, if I print out what that looks like, it looks like this. I have a data frame that has 3 different columns. And so what I'm interested in doing is interested in the height specifically. I'm not interested in this column or this column. I just want the heights. So how do I access that? Well, I can write the name of the variable, which is students in this case, student heights. And I want just that middle column. And so I can choose with a $sign, I choose height. I press enter, and now I just get the heights, okay? And that's what I wanted. Now, what would I like to do? What's something we'd like to compute about the heights? Well, let's compute the average or the mean. How would we do that? Well, it turns out for all these functions, it's just often what you'd expect. So to compute the mean, you write mean. And now we have to specify what do we want to compute the mean of. We want to compute the mean of the heights. So this is the way we have to write it here. And now it's going to return that value for me. If I wanted to compute the median, what would be the name of the function that you think would compute the median?
Median.
Correct. So I can now just write median instead, and I get that value. Now, one way to do this now is to take these things that we know work, these commands, go up here, insert a new, and type that here. And now the way that in Markdown, the way I'm going to run that is I'm going to press this little green button here, and now it's going to print my mean right here. I could also I know the median works. I can go up here, and I can do the same thing, and if I press it again, it's going to compute both of them, okay? And now I know we're a few minutes over. If you can give me 5 more minutes, here's the key thing I want to show you how to do, is I would like to plot a histogram of the heights. And I spelled heights wrong, but that's fine. So how am I going to do that? I'm going to insert a new chunk here. And if I want to plot a histogram, there's a function for that. Okay, what do you think the function for plotting a histogram would be called? What's a good guess?
Plot histogram.
Plot histogram, those are all possibilities. And so often the challenge is, what is the function that draws a histogram in R? And what would be a good way to figure that out?
Type histogram.
Yeah, you could do that. And the practical way, and the way that I would do it, is I would just go to Google and say, how do you plot a histogram in R? You know, until you know these things, that's a lot of what we do. Is if we want to do something that we don't really know how to do, you can ask in R, because it turns out that the function is actually hist. And so now I want to remember, just to get that column of values, I've used this syntax. So now if I plot just hist, I'm going to get a histogram. Very nice. Now, what did I tell you? Every histogram must always have a x-axis and a y-axis. In this case, the x-axis is not very useful. So I want to add an x-axis, so I need to add an argument to this, and that's xlab, xlabel. And that is going to be student heights. I cannot spell heights, in centimeters. Okay, so now what happens? I have my x-axis label, and that works nicely. I prefer it to be blue. What do? I can add another argument to this same thing, and I can call now the col, short for color, equals blue. I can try and run that. What happens? It's blue. Okay, and so I don't like this title, perhaps, so now I need to change that. I'm going to say the main equals, I have to specify, this is the syntax it's called, the main equals student heights in 2021 biostats. Stats. Is that correct? Spell? Yeah. And now I have a nice height. Okay. I could do the same plot now let's say I'm going to insert another chunk and I wanted to do the same thing, but as a box plot, which I told you about. Now we can do student heights. We're going to take just the height column by using this syntax, and I'm going to run that. And now I have a box plot. But it has a fundamental problem, which is that it doesn't have a Y label. And so your job is going to be add ylab and add it, okay? So we're over time. I will complete this and I'll post this .rmd file on classes so you can take a look at it. And but I just wanted to run you through, you know, how I do things and show you the key points, which is that you need to know where you are. You need to know how to import things, and often you'll have problems importing data. That's often, if I had those missing values that I hadn't filled in, I might have had a problem. So that's often a problem. And to interact with R, I can either interact with it down here in this console, but none of that will be saved, or I can interact with it up here in this, using R Markdown. And now when I press preview, it's going to generate a nicely formatted document that has all my code and all the output on it. And that's what you're going to be doing in this class, okay? All right. Apologies for going over time. I think, let's see, ask Google, that's correct, that you'll be doing that a lot. All right. I don't have to rush off. I'll stop sharing here. I'll stop recording now, but if anyone wants to stay and ask any questions, I'm free to stay around for a few minutes as well. So